So we're going to wrap up our Chapter 15 Principles of Metabolic Regulation by continuing to look at the regulation of phosphofructokinase 1 and fructose 1,6-bisphosphate. So we go glycolysis if AMP is high and ATP is low. We go gluconeogenesis if AMP is low. And so this really shows the important role of fructose 2,6-bisphosphate in the regulation of the substrate cycle. If you look at fructose 2,6-bisphosphate, it's not a glycolytic intermediate, it's only a regulator. So it's produced specifically to regulate glycolysis and gluconeogenesis. It activates phosphofructokinase for glycolysis, and it inhibits fructose 1,6-bisphosphatase for gluconeogenesis. Because glycolysis and gluconeogenesis are actually differentially regulated by phosphofructokinase 2,6-bisphosphate, by fructose 2,6-bisphosphate. When we're looking at this, really it's the fructose 2,6-bisphosphate that has opposite effects on the enzyme activities. So if I'm looking at PFK1 activity in the absence of fructose 2,6-bisphosphate, that's the blue curve. It's going to be at half maximal when the concentration of fructose 6-phosphate is 2 millimolar. When I have 0.13 micromolar fructose 2,6-bisphosphate present, the red curve, right, then my fructose 6-phosphate is only about 0.08 millimolar. So my fructose 2,6-bisphosphate activates PFK1 by increasing its apparent affinity for fructose 6-phosphate. And then if I look at fructose bisphosphatase 1 activity, that's going to be inhibited by as little as 1 micromolar. And it's strongly inhibited by 25 micromolar. So in the absence of this inhibitor, the blue curve, fructose 1,6-bisphosphate is 5 micromolar, but in the presence of 25 micromolar, fructose 2,6-bisphosphate is going to be less than 0.7 micromolar. So this also makes the fructose bisphosphatase 1 more sensitive to inhibition by another allosteric regulator, AMP. We can actually see that here. So this is this is the impact of the hydrolysis of ATP and the production of fructose 2,6-bisphosphate. So if I'm looking at regulation of 2,6-bisphosphate, I can actually see here that, right, my concentration stimulates glycolysis and can inhibit gluconeogenesis. If my concentration of fructose 2,6-bisphosphate goes down, that inhibits glycolysis and stimulates the gluconeogenesis. So allosterically activated fructose 1,6-bisphosphate is a high flow through glycolysis. It allosterically inhibited by signs of abundant energy supply. So if I have lots of ATP, if I have acetyl-CoA and long-chain fatty acids, or alanine, right, that tells me I have enough amino acids, it's going to be inactivated by phosphorylation in response to signs of glucose depletion. So if there's glucagon or in the liver only. So glucose from the liver is exported to the brain and other vital organs. If I'm looking at the regulation of pyruvate kinase, this happens in all glycolytic tissues, including the liver versus in the liver only. So this enzyme is allosterically inhibited by ATP, acetyl-CoA, and long-chain fatty acids, all signs of abundant energy. And the accumulation of fructose 1,6-bisphosphate triggers its activation. So accumulation of alanine, which can be synthesized from pyruvate in one step, allosterically inhibits pyruvate kinase, slowing the production of pyruvate by glycolysis. If I'm looking at the liver isozyme L-form, I can also regulate that hormonally. So glucagon activates CAMP-dependent protein kinase, which is going to phosphorylate the pyruvate kinase L isozyme and activating it. So when the glucagon levels drop, a protein phosphatase dephosphorylates pyruvate kinase, activating it. This is going to prevent the liver from consuming glucose by glycolysis when blood glucose is low. Instead, the liver exports glucose. The muscle isozyme M-form is not affected by this phosphorylation mechanism. So what does that mean? Well, I have two alternative fates for pyruvate. Pyruvate can be a source of new glucose. It can actually store energy as glycogen and generate NADPH via the pentose phosphate pathway. Pyruvate can also be a source of acetyl-CoA. It can store energy as body fat. It can make ATP via the citric acid cycle. And acetyl-CoA actually stimulates glucose synthesis by activating pyruvate carboxylase. If I'm looking at the alternative fates for pyruvate, I can convert it to glucose and glycogen via gluconeogenesis, or oxidize to acetyl-CoA for energy production. The first enzyme in each pathway is regulated allosterically. Acetyl-CoA is produced either by fatty acid oxidation or by the pyruvate dehydrogenase complex, which stimulates pyruvate carboxylase and inhibits pyruvate dehydrogenase. So the amount of most of these metabolic enzymes is going to be controlled by transcription, and you can actually see the gene role here. So genes have this ability to impact transcription in response to glucose. Right? I can see activation of transcription in response to insulin. And I can also, right, control the transcription of genes by a variety of factors. When I think about glucose residues that are removed from glycogen by glycogen phosphorylase, right, the idea here is that glycogen phosphorylase works on non-reducing ends until it reaches 4 residues from an alpha 1,6 branch point. So this debranching enzyme transfers a block of 3 residues to the non-reducing end of the chain, and it also cleaves a single remaining alpha link to glucose. We've also seen other examples of regulation in the form of epinephrine and glucagon to stimulate the breakdown of glycogen. And we can actually control glycogen synthesis via the insulin signaling pathway, which increases glucose import into muscle, stimulates the activity of the muscle hexokinase, and activates glycogen synthase. So increased hexokinase activity enables the activation of glucose, and glycogen synthase makes glycogen for energy storage. These are all some of the ways that we can control metabolic pathways. Thanks again.